Welcome to Cosmic Whispers, where cosmic mysteries come alive. Your journey beyond the stars begins now. In the azure depths of the Mediterranean Sea, a transparent bell-shaped creature, no larger than a human fingernail, drifts with the currents. To casual observers, it appears unremarkable, just another of the countless jellyfish species that have inhabited Earth's oceans for over 600 million years. But this particular organism, Turritopsis dohrnii, harbors an extraordinary biological secret that has captivated scientists worldwide. It possesses the ability to reverse its life cycle and return to youth after reaching maturity. The discovery of this remarkable creature in the late 1980s sent ripples through the scientific community. While all other known species journey inevitably toward death, Turritopsis dohrnii can, when injured or environmentally stressed, revert to an earlier developmental stage. Imagine a butterfly transforming back into a caterpillar or a human adult. Reverting to childhood, biological impossibilities for nearly every multicellular organism except this unassuming jellyfish. Biologists use the term biological immortality to describe this phenomenon, though it doesn't mean the jellyfish cannot die. Rather, it possesses a theoretical immortality. If it can avoid predators, disease, and other external threats, it could potentially live indefinitely by continuously resetting its biological clock. This capacity represents one of the most extreme forms of regeneration in the animal kingdom. Every great journey starts with a few brave explorers. Right now, Cosmic Whispers is just beginning, and your support means everything. If you enjoy unraveling the mysteries of the universe, hit subscribe. It's free, and it helps us grow this community together. The process through which Turritopsis dohrnii achieves this remarkable feat is called transdifferentiation, a cellular alchemy where specialized cells can transform into entirely different cell types without passing through an embryonic or stem cell state. When the jellyfish enters its rejuvenation process, its cells undergo a profound reorganization, dismantling its mature structures and rebuilding itself as a polyp, the earliest stage of jellyfish development. Dr. Stefano Pirino of the University of Salento in Italy, one of the pioneers in studying this species, describes it as a butterfly that instead of dying, shrinks into a caterpillar to begin life anew. This capacity isn't just regeneration, it's a complete biological reset that effectively renders the organism young again. The scientific significance cannot be overstated. In a universe governed by entropy, where everything tends toward disorder and decay, Turritopsis dohrnii represents a biological anomaly, a creature that has evolved to temporarily reverse entropy's arrow at the cellular level. It stands as a living challenge to the seemingly universal progression from birth to growth to decline and death. Since its discovery, this tiny hydrozoan has earned numerous monikers in scientific literature and popular media. The Immortal Jellyfish, the Benjamin Button of the sea, and the Phoenix of the ocean. These dramatic titles reflect both the extraordinary nature of its biology and humanity's perennial fascination with immortality. As we delve deeper into the molecular mechanisms behind this remarkable creature's abilities, we find ourselves exploring one of biology's most profound questions. Is aging an inevitable process, or simply a particular evolutionary path that some organisms have found ways to circumvent? The answer may reshape our understanding of life's fundamental processes and, perhaps, offer insights into our own mortality. To appreciate the extraordinary nature of Turritopsis dohrnii's immortality, we must first understand the typical jellyfish life cycle. Most jellyfish begin life as a fertilized egg that develops into a free-swimming planular larva. This microscopic larva eventually settles on a suitable surface and transforms into a polyp, a stationary plant-like structure. The polyp then produces and releases multiple medusae, the bell-shaped organisms we recognize as jellyfish, which grow to sexual maturity and reproduce, completing the cycle. For most jellyfish species, the medusa stage represents the culmination of their existence. After reproducing, they deteriorate and die, leaving their genetic legacy to the next generation. This progression from birth to reproduction to death follows the standard pattern observed throughout the animal kingdom, a one-way journey through developmental stages that culminates in senescence and mortality. Turritopsis dohrnii follows this typical pattern, but with a crucial difference. When faced with environmental stress, physical injury, starvation, or even old age, the mature medusa has the capacity to revert to the polyp stage. Dr. Maria Pia Milieta, a marine biology professor at Texas A&M University, explains, "This would be like a butterfly that transforms back into a caterpillar, or a frog that reverts to a tadpole, or a human that reverts to a fetus. It's a complete reversal of the normal life trajectory." During this remarkable transformation, the jellyfish's bell begins to collapse. Its tentacles are reabsorbed, and its cells undergo transdifferentiation, a process where one cell type converts to another without passing through an undifferentiated stem cell phase. The entire organism then settles on a surface and transforms into a polyp colony, essentially becoming young again. This new polyp colony can then produce new genetically identical medusae, which can themselves mature, reproduce, and potentially undergo the same rejuvenation process. This biological loop creates a theoretical immortality. While predators, disease, or accidents can still terminate its existence, Turritopsis dohrnii has evolved a mechanism to escape what biologists call programmed cell death, or apoptosis, the genetic instructions that limit cellular divisions and lead to aging in most organisms. The cellular mechanisms behind this process involve complex interactions between environmental stimuli, genetic regulation, and cellular reprogramming. When the jellyfish initiates its rejuvenation sequence, it activates genes associated with development and growth, while simultaneously suppressing genes linked to maturity and senescence. This genetic reprogramming cascades through the organism, triggering widespread cellular transformations. Research conducted at the University of Lecce in Italy has revealed that during this process, muscle cells can convert into nerve cells can become egg cells, and specialized digestive cells can transform into entirely different tissue types. This cellular plasticity, the ability of differentiated cells to transform into other cell types, represents an extraordinary example of biological flexibility rarely seen in multicellular organisms. What makes this ability particularly remarkable is that it occurs in an animal that already possesses a nervous system, digestive tract, and other complex biological structures. Most organisms with high cellular plasticity are relatively simple, but Turritopsis dohrnii demonstrates that even creatures with sophisticated body plans can maintain the genetic programming necessary for complete biological reinvention. At the molecular level, the immortality of Turritopsis dohrnii involves sophisticated genetic and cellular mechanisms that researchers are only beginning to unravel. In 2013, scientists from Japan's National Institute of Advanced Industrial Science and Technology sequenced the genome of this remarkable organism, providing the first comprehensive look at the genetic architecture behind its age-defying abilities. The genome revealed several interesting features, including an abundance of genes associated with DNA repair, antioxidant production, and stem cell regulation, all critical components in combating cellular aging. Particularly notable was the discovery of enhanced copies of the FOXO gene, which plays a crucial role in stress resistance and longevity across many species, including humans. When most animals experience cellular damage, whether from environmental, toxins, metabolic byproducts, or simple wear and tear, they activate repair mechanisms to fix the damage. If these mechanisms fail, the damaged cells typically enter senescence, a state where they no longer divide, or undergo apoptosis, programmed cell death. This ongoing loss of functional cells contributes significantly to aging. Turritopsis dohrnii, however, possesses an expanded toolkit for cellular maintenance. Its enhanced DNA repair systems can fix damage that would be permanent in other organisms. More remarkably, when damage becomes too extensive, rather than allowing cells to deteriorate, it initiates transdifferentiation, essentially recycling the cellular material into younger, healthier cells. Dr. Shin Kubota from Kyoto University, who has maintained colonies of these jellyfish in his laboratory for over 15 years, has observed their rejuvenation process hundreds of times. The process appears to be triggered by environmental stresses, he explains. When conditions become unfavorable, rather than struggling to survive in a compromised state, they revert to an earlier developmental stage where they can essentially start over. The silencing and activation of specific genes orchestrate this remarkable transformation. Genes associated with medusa development become suppressed, while genes linked to polyp formation activate. This genetic reprogramming cascades through the organism, triggering widespread cellular transformations that collectively reorganize the entire body plan. Telomeres, the protective caps at the ends of chromosomes that shorten with each cell. Division in most organisms also play a role in this process. In humans and many other species, telomere shortening serves as a molecular clock that eventually signals cells to stop dividing, contributing to aging. Studies suggest that Turritopsis, dohrnii, maintains robust telomerase activity, the enzyme responsible for rebuilding telomeres, throughout its life cycle, effectively resetting this molecular clock during rejuvenation. The jellyfish also exhibits remarkable control over its epigenome, the chemical compounds and proteins that can attach to DNA and direct actions like gene expression. During transdifferentiation, extensive epigenetic reprogramming occurs, silencing genes associated with the medusa stage while activating those linked to polyp development. This epigenetic flexibility allows cells that have been specialized for one function to adopt entirely new identities. Perhaps most intriguing is the jellyfish's apparent lack of the Waddington landscape constraints that limit cellular plasticity in most complex organisms. This theoretical framework, proposed by British biologist Conrad Waddington, describes how cells become increasingly restricted in their developmental potential as they specialize. Turritopsis dohrnii seems to possess mechanisms that allow it to overcome these constraints, enabling mature specialized cells to revert to less differentiated states. The evolutionary path that led to Turritopsis dohrnii's remarkable ability presents a fascinating puzzle for biologists. While the capacity to reverse aging would seem to confer an overwhelming survival advantage, this trait has not become widespread in the animal kingdom. This paradox raises profound questions about the evolutionary trade-offs involved in biological immortality. Dr. James Carlton, professor of marine sciences at Williams College, suggests that immortality may come with hidden costs. Natural selection favors traits that enhance reproductive success, not necessarily longevity. If the energy investment required for transdifferentiation comes at the expense of reproductive output or other survival traits, then immortality might not be as advantageous as it first appears. The evolutionary history of Turritopsis dohrnii likely involves a series of adaptations originally meant to enhance survival during environmental stress. Many marine invertebrates possess remarkable regenerative abilities, and some cnidarians, the phylum that includes jellyfish, can revert certain body parts to earlier developmental stages when injured. The immortal jellyfish appears to have extended this capacity to encompass the entire organism. Genetic studies comparing Turritopsis dohrnii with its mortal relatives have identified several key differences in genes related to stem cell regulation, DNA repair, and cellular reprogramming. These genetic innovations likely accumulated gradually over millions of years, ultimately conferring the unique ability to reset its life cycle completely. From an ecological perspective, biological immortality creates interesting population dynamics. Since 2010, researchers have observed Turritopsis dohrnii spreading to oceans worldwide, far beyond its native Mediterranean habitat. Marine biologists have documented its presence in the waters of Japan, Spain, Florida, Panama, and elsewhere, an expansion facilitated by ballast water in international shipping vessels. This global distribution has raised concerns about potential invasive species impacts. Dr. Maria Milieta notes that a species that can potentially live forever and reproduce both sexually and asexually presents unique challenges from a management perspective. While no negative ecological impacts have been conclusively documented, the potential for rapid population growth exists, if environmental conditions prove favorable. Unlike most invasive species, which typically require large founding populations to establish themselves in new environments, Turritopsis dohrnii can potentially establish a new population from a single individual. Through asexual reproduction during its polyp stage and its ability to revert to this stage when stressed, even a lone jellyfish can theoretically generate a sustainable colony. Their ecological resilience appears particularly relevant in today's changing oceans. As climate change alters marine environments through warming waters, changing currents, and acidification, many species face significant challenges. Turritopsis dohrnii's biological flexibility may provide an advantage in these shifting conditions. Rather than requiring generations of natural selection to adapt, individual organisms can effectively reset and potentially adjust to new environmental parameters. This adaptability raises important questions about the future distribution of these jellyfish. Models predicting marine ecosystem changes suggest that many cnidarian species may benefit from warming oceans and increasing coastal development. Some marine biologists speculate that highly adaptable species like Turritopsis dohrnii could become increasingly common in disturbed marine environments. Despite these concerns, tracking wild populations remains challenging due to the jellyfish's small size, transparency, and morphological similarity to related species. Our understanding of their natural abundance, distribution, and ecological role continues to evolve as research advances. In laboratories around the world, researchers maintain colonies of Turritopsis dohrnii to study their remarkable biology under controlled conditions. These scientific outposts serve as windows into the mechanisms of biological immortality, with each rejuvenation cycle meticulously documented and analyzed. Dr. Shin Kubota's laboratory at Kyoto University has maintained the longest continuous culture of these jellyfish, observing hundreds of rejuvenation cycles over more than 15 years. His work has demonstrated that while the process occurs naturally in response to injury or stress, it can also be induced through careful manipulation of environmental conditions, providing researchers with a reliable experimental system. 1. Of the most significant breakthroughs came in 2016, when an international team of researchers successfully mapped the complete gene expression profile of Turritopsis dohrnii throughout its life cycle, including during the critical transdifferentiation phase. This transcriptomic analysis revealed cascades of genetic activity that coordinate the rejuvenation process, identifying key regulatory genes that act as molecular switches between developmental stages. Particularly noteworthy was the identification of increased expression of genes associated with pluripotency, the ability of cells to develop into different cell types. These genes, including homologs of OCT4 and SOX2, are typically active during early embryonic development in most animals, but become silenced in mature tissues. In Turritopsis dohrnii, they can be reactivated in adult cells, effectively reprogramming them to an earlier developmental state. The research also revealed sophisticated epigenetic regulation mechanisms. During transdifferentiation, extensive chromatin remodeling occurs, the physical restructuring of how DNA is packaged within cells. This remodeling makes previously inaccessible genes available for activation, while silencing genes associated with the Medusa stage. This epigenetic plasticity appears crucial for the drastic cellular transformations observed. Beyond genetics, researchers have made significant advances in understanding the biophysical aspects of the transformation. Using advanced imaging techniques like confocal microscopy and electron tomography, scientists have visualized the dramatic cellular reorganization that occurs. These studies reveal how muscle cells disassemble their contractile fibers, epithelial cells lose their specialized junctions, and neural cells retract their processes as the organism prepares to rebuild itself from the cellular level up. Biochemical analysis has identified several signaling molecules that appear to coordinate this transformation, including Wnt proteins, bone morphogenetic proteins, BMPs, and transforming growth factor beta, TGF-beta. These molecular signals, which guide embryonic development in many animals, appear to be redeployed during transdifferentiation to orchestrate the rejuvenation process. The cellular energetics of transdifferentiation have also received attention. Researchers at the University of Copenhagen have demonstrated that the transformation requires a significant metabolic shift, with cells temporarily increasing mitochondrial activity to generate the energy needed for wholesale cellular reorganization. This metabolic flexibility may be another key adaptation enabling the jellyfish's remarkable life cycle. Perhaps most intriguing are recent experiments exploring the limits of this rejuvenation capacity. Dr. Kubota's work suggests that while theoretically immortal laboratory colonies eventually show signs of diminished vigor after multiple rejuvenation cycles, what he terms rejuvenation exhaustion. This phenomenon hints at currently unknown constraints on biological immortality, possibly involving accumulated damage to mitochondrial DNA or other cellular components that escape the rejuvenation process. Despite these limitations, research continues to expand our understanding of how Turritopsis dohrnii cheats death. Each new insight not only illuminates the biology of this remarkable organism, but also provides potential applications for human health and longevity research. The medical implications of Turritopsis dohrnii's abilities have sparked intense interest among researchers studying human aging and regenerative medicine. While humans will never reverse their life cycle like these jellyfish, specific cellular mechanisms involved in transdifferentiation may offer valuable insights for treating age-related diseases and injuries. Cellular reprogramming, converting one cell type into another, represents one of the most promising applications. In 2012, Japanese scientist Shinya Yamanaka received the Nobel Prize for discovering that mature human cells could be reprogrammed into induced pluripotent stem cells, iPSCs, capable of developing into various cell types. This breakthrough, which revolutionized regenerative medicine, shares conceptual similarities with the jellyfish's natural abilities. Dr. Maria Blasco, director of the Spanish National Cancer Research Center's Molecular Oncology program, explains the connection. The jellyfish achieves naturally what we're attempting to do therapeutically, reprogramming cells to restore youthful function. Studying their mechanisms could reveal more efficient or safer methods for cellular rejuvenation in human tissues. Particularly relevant are the jellyfish's controlled activation of pluripotency factors. In human therapeutic applications, excessive activation of these factors can lead to cancer, while insufficient activation fails to achieve reprogramming. Turritopsis dohrnii has evolved precise regulatory mechanisms that activate these factors just enough to enable transdifferentiation without triggering uncontrolled growth, a balance researchers are eager to understand and potentially replicate. Telomere biology represents another promising area of crossover research. In 2019, researchers at the University of Oviedo in Spain identified similarities between telomere maintenance mechanisms in the immortal jellyfish and those being investigated. For human anti-aging therapies, their work suggests that certain enzymatic pathways involved in the jellyfish's telomere preservation might be adaptable to human cells to combat cellular senescence. Regenerative medicine applications extend beyond aging research. Scientists at Harvard's Weiss Institute have drawn inspiration from the jellyfish's cellular plasticity to develop improved methods for tissue engineering. By studying how Turritopsis dohrnii cells communicate during transdifferentiation, they've identified signaling molecules that could potentially enhance tissue regeneration in human organs damaged by disease or injury. The jellyfish's extraordinary stress resistance mechanisms also offer potential applications. Research at the Buck Institute for Research on Aging has identified parallels between stress response pathways in the immortal jellyfish and those that, when activated in laboratory, mammals increase healthy lifespan. These pathways involve cellular quality control systems that remove damaged proteins and organelles, processes that decline with age in humans but remain robust in Turritopsis dohrnii throughout its life cycle. Cancer research represents yet another frontier where insights from the immortal jellyfish prove valuable. The organism's ability to control cell proliferation during transdifferentiation, activating growth pathways without triggering tumor formation, could inform new approaches to cancer treatment. Scientists at Memorial Sloan Kettering Cancer Center are investigating how the jellyfish's regulatory mechanisms might be adapted to suppress uncontrolled cell division in human cancer cells. Pharmaceutical companies have taken notice of these potential applications. Several biotech firms have launched research programs specifically focused on translating insights from Turritopsis dohrnii biology into therapeutic interventions. These efforts range from developing small molecule compounds that mimic the jellyfish's cellular protection mechanisms to creating genetic therapies inspired by its regenerative pathways. While practical applications remain largely experimental, the conceptual shift inspired by the immortal jellyfish has already influenced medical research. By demonstrating that biological aging can be reversed at the cellular level, Turritopsis dohrnii has helped legitimize the view that aging itself might be treated as a medical condition rather than an inevitable process, a perspective that continues to gain traction in biogerontology. The immortal jellyfish challenges our deepest assumptions about the nature of life and death. For millennia, human cultures worldwide have recognized linear progression from birth to death as a fundamental law of existence. Turritopsis dohrnii, with its circular life cycle, offers a biological counterargument to this seemingly universal truth. This biological reality raises profound philosophical questions. If aging is not biologically inevitable, but rather one evolutionary strategy among many, how should we understand our own mortality? Dr. Aubrey de Grey, a biomedical gerontologist and chief science officer of the SENS Research Foundation, suggests that the jellyfish demonstrates that aging is not an intrinsic property of life, but rather a particular developmental path. This realization fundamentally changes how we should approach human longevity research. The immortal jellyfish also challenges our understanding of individuality and identity. When Turritopsis dohrnii reverts to a polyp and subsequently produces multiple genetically identical medusa, is the resulting organism the same individual that underwent rejuvenation or something new? This question parallels philosophical puzzles like the Ship of Theseus, which asks whether an object that has had all its components replaced remains fundamentally the same object. Looking toward the future, research on biological immortality continues to accelerate. Advanced genetic technologies like CRISPR-Cas9 now allow scientists to precisely modify genes in various organisms, potentially transferring specific rejuvenation mechanisms from Turritopsis dohrnii to experimental models. While no one expects humans to ever undergo complete life cycle reversal, the transfer of specific cellular mechanisms could substantially impact medicine. Dr. João Pedro de Magalhães, who leads the Genomics of Aging and Rejuvenation Lab at the University of Liverpool, believes that comparative biology approaches offer particular promise. By studying species with exceptional longevity or regenerative capabilities, we can identify conserved pathways that might be targeted therapeutically in humans. The immortal jellyfish represents an extreme case that may reveal fundamental principles of cellular rejuvenation. The ethical dimensions of this research also merit consideration. As our understanding of biological aging improves, questions about appropriate applications become increasingly important. Should we prioritize extending maximum human lifespan or focus on health span, the portion of life spent in good health? How would dramatically extended lifespans affect social structures, resource allocation, and environmental sustainability? The jellyfish, with its theoretical immortality, provides a biological reference point for these otherwise abstract discussions. Environmental considerations add another layer to this conversation. As marine ecosystems face unprecedented pressures from climate change, pollution, and overfishing, understanding organisms that display exceptional resilience becomes increasingly important. The adaptability demonstrated by Turritopsis dohrnii might offer insights into how organisms respond to environmental stress, knowledge that could prove valuable for conservation efforts. The immortal jellyfish also reminds us of how much remains to be discovered in Earth's oceans. Despite centuries of scientific inquiry, marine biologists estimate that up to 2/3 of marine species remain undescribed. Each of these unknown organisms represents a unique evolutionary experiment, potentially harboring biological innovations, as remarkable as the immortality of Turritopsis dohrnii. As we continue exploring the biological frontiers of aging and regeneration, the immortal jellyfish serves as both inspiration and guide. Its extraordinary capabilities remind us that nature's innovations often exceed our imagination, while its biological mechanisms provide concrete pathways for scientific investigation. In the depths of the world's oceans, this small, transparent creature continues its ageless drift, embodying one of nature's most remarkable solutions to the challenge of survival, not by defeating death, but by incorporating it into a continuous cycle of renewal.